Okay, the second speaker is Dr. Alfred Alberts, and Dr. Alberts is a pharmaceutical and scientific consultant specializing in drug discovery and development. He retired after 20 years from Merck Research Laboratories in 1995, where his most recent position was Vice President of Natural Product Discovery. While at Merck, he directed programs in atherosclerosis, diabetes, and obesity. He was responsible for the discovery and development of Mevacor and Zocor. Prior to joining Merck, he was a faculty at the Department of Biochemistry at Washington University School of Medicine, where he rose from research instructor to associate professor of biochemistry. Mr. Alberts is the author of 6 patents, including 3 for the discovery of LOVASTATIN, as well as over 75 publications. And Dr. Alberts.
Thank you, Pat, and let's hope there's no glitches here. You got them all. First slide, please. I think, I can do that, right. On the first slide, I think really summarizes one of the big issues that Joe discussed. Much of this is the big fear, and when you develop a drug. You have a breakthrough, you think you have something based on animals, on various test systems, you put them into man, and what happens? You get a placebo effect for what you're looking for, but you have the side effects. And that costs a lot of bucks. For the big companies, what that means is you can bury the project in the middle of the night, nobody knows about it. For the little biotech companies, the stock goes from $10 to 10 cents overnight. Big companies, they say, can bury that. Now, we had a program that was very different. Here's our objectives, really summarized on this slide, and what we had to do to obtain these objectives. Cholesterol, triglycerides, and hot air are too high. Our objective was to discover a drug to lower cholesterol. In the process, if we came up with something that had triglyceride effect, great, but that wasn't what we were looking for. And to get this through and to really develop it, we had to overcome a lot of hot air, both internally and externally. And I think Joe knows what I'm talking about. This is dealing with marketing, it's dealing with competitors, and Merck was one of the competitors for Joe, and now Pfizer is one of the competitors for Merck. But really, the basis of our work is shown here. It's based on the lipid hypothesis, lowering serum cholesterol, logical intervention to limit progression or cause regression of coronary atherosclerosis, and in turn, of course, coronary heart disease. Coronary heart disease is still the major cause of death in the Western world, with all the nice new drugs out there. And one of the ideas was that high cholesterol was the responsible for a lot of coronary atherosclerosis. Now, cholesterol is always thought of as being some bad player. However, it is only a bad player when there's too much. And too much means too high a level in the circulation. Cholesterol structure shown here is a 27-carbon steroid that's involved in the key synthesis of a lot of your steroid hormones. It is involved in transport of lipids through the body. It's involved in absorption of lipids. And it is also the key component of all mammalian cell membranes. Without cholesterol, your membranes don't exist. So it's important. Our objective then was not to eliminate cholesterol, but modulate its production. Now, where does cholesterol come from in the body? It comes from 2 sources. One, the obvious one that everybody knows about, the diet. Eggs, milk, dairy products, meat, etc. However, there is more cholesterol made within the body than it is absorbed from the diet. So just lowering intake of cholesterol often proves inadequate in lowering cholesterol levels to a level that is sufficient to prevent atherosclerosis. So I'm going to talk about two molecules, LOVASTATIN and SYMVASTATIN, Mevacor and Zocor. Joe mentioned the history at Pfizer for Feldane. There was a long history at Merck and outside of Merck on looking for compounds to lower cholesterol. And I could trace Merck's early involvement in this back to 1956 when a molecule known as mevalonic acid was isolated. The purpose of this discovery, this was isolated from yeast extract. This was at a time when vitamins were the big thing, and at Merck they were looking for novel sources of vitamins, and mevalonate was going to be used as a food additive for chickens to increase the growth of chickens. It had no effect on growth of chickens. But there was somebody else at Merck at the time that was studying cholesterol synthesis, and he noticed this resemblance of mevalonic acid to an intermediate in cholesterol biosynthesis, and actually was first demonstrated that this molecule was important and was a key intermediate in cholesterol synthesis. Shortly thereafter, the enzyme that made mevalonic acid was discovered. That's HMG-CoA reductase, and that is the basis of what I will be talking about later. And this was shown to be the enzyme that controlled how much cholesterol was being made in the body. It was the rate controlling enzyme. It's the enzyme that really, if the enzyme is very active, you'll get a lot of cholesterol made. If it's not very active, you'll get a little bit of cholesterol made. Another key event at that time was a compound known as cholestyramine. It's marketed now as Questran. This is a compound that blocks the reabsorption of bile acids from the intestine and does work to lower cholesterol. Very unsatisfactory compound in terms of properties. It looks, well, in those days it was even worse than now. It smelled like rotten fish, and it tasted like sand. Now they've gotten rid of the fishy smell. It still has a taste like sand, but it is an effective compound. That too was discovered at Merck, but Merck could never formulate it in a way to market it, and they licensed it out to Mead Johnson, which is now really Bristol Myers Squibb, and they do market the compound. In the 1970s, it was shown that you could regulate the level of HMG-CoA reductase, and Merck instituted a limited screen to find new compounds that worked by this mechanism. The key event that occurred, unfortunately not at Merck, was the discovery of a compound in Japan, which went by several names, Mevastatin, Compactin, and that was by Endo at Sankyo, and I'll have a little more to say about that. This was a natural product isolated from a fungus, Penicillium citrinum. And then in 1977, we initiated a program at Merck to discover novel analogs of this compound. Very few companies, including Pfizer and Merck and all, come up with novel ideas for drug targets. Everybody follows the history, follows what's already known, and tries to come up with new, improved molecules, and not necessarily me-toos, but they're new structures that may or may not do similar things as old molecules. And we initiated screening of extracts in 1978, and as I will show you, rapidly came up with a new entity, LOVASTATIN, again from another fungus, Aspergillus terreus. Now, just to show a little biochemistry, and I'm a biochemist, not a chemist, so we have to have some of this done. This just shows a very brief outline of the cholesterol biosynthetic pathway. And I show it because several important points here. One is that there are many steps, and I've just not indicated all of them. There are more than 25 separate distinct enzymes involved in cholesterol biosynthesis. The one that we have focused on for a number of reasons, as I indicated, is the HMG-CoA reductase, which involved in the conversion of a molecule. Is this working? The reaction known as hydroxymethylglutaryl coenzyme A, HMG-CoA, in 2 steps, single enzyme, to mevalonic acid. And as I indicated, mevalonic acid is the key intermediate in cholesterol biosynthesis. We used this reaction as part of our test system. Enzyme was partially purified from rat liver. This was pre-recombinant enzymes. Subsequent to this work and later parts of our work, we did use recombinant human HMG-CoA reductase. It was easier to handle than the rat enzyme, but the rat enzyme behaved identically to the human enzyme, fortunately for us. We set up a screen based on starting with radioactive HMG-CoA and isolating radioactive mevalonic acid. Very simple screen. For its day, it was a very high throughput assay. We could have one person, my colleague Julie Chen, actually set this up, and she personally was able to run about 200 assays in a day by herself with no help. Very high throughput for 1978. Well, we set this up with the idea that we were going to have a lot of compounds coming through. We had a chemical program underway. The chemical program was, for me, being very impatient, was going too slowly, and not enough compounds were coming through to generate enough work for this screen that we could do 200 in a day. I think the compounds we would get about, if we were lucky, 5 a week. So we had another source of material at the time, and that was extracts of natural products. Art Patchett at Merck, who is the chemist at Merck that was responsible for the drug enalapril, and also ran the isolation group, had the idea that this could be done in Japan. Why couldn't it be done in the States, that we look at natural products? And he set up a very small scale approach initially. And what he did was isolate microorganisms from various ecological niches. These organisms were fermented in different media, extracted, and tested for inhibitory potential, and then you go and isolate the pure compound and then go through a scale-up to produce large quantities of a compound. That's the standard protocol. This was a protocol basically adapted from the antibiotic programs that were underway, and we did this. We used this as a source of material. At the time, I said we were impatient with the chemical approach. All we were getting were 15 to 20 extracts a week. The first week we got 17. Sample number 18 was a fungus shown on this. This is my favorite organism, Aspergillus terreus, common soil microorganism, which produced in high yield a compound shown on the bottom, lovastatin, which is shown here as the active form of the molecule. As I show you later, the compound is not sold in this form, but with the closed ring on top as a lactone. And I won't go into the reasons for it now because of time, but this compound was a potent is a potent inhibitor of the target enzyme, HMG-CoA reductase. Now, you'll notice on the top part of the lovastatin molecule the close resemblance to the substrate and the product of the reaction. You have a carboxylic group, you have a hydroxyl, if it's the carboxyl is number 1, and next is a hydroxy in number 3. There's also a methyl in number 3, a mevulanic acid that is missing from lovastatin. It's not in lovastatin. I should say that if that methyl group was put into, chemically introduced into lovastatin, and one would expect improved activity from that, and instead there was loss of activity rather than improved. Yes?
Were you doing some chemistry around mevulanic acid at that time also? Or?
We were doing chemistry around the basic structure of the molecule. We were not doing chemistry around mevulanic acid.
Why not?
Because there had been a lot of that work done in the past with no success. The I have to say that here we came up with lovastatin, and I mentioned another compound, mevastatin, ML236B. It differs from lovastatin only by a methyl group. Has one less methyl group. That was the basis for other chemistry work going on at Merck. Mevulanic acid did not prove to be a good molecule to work on.
But at that time, when you were screening your natural library, natural compound library, you knew of mevastatin.
Yes.
So why weren't you doing some chemistry around mevastatin?
We were. And that's what was going, I mean, I'll mention a little more about that when I go on with the not directly around that, but when I go on with what was done subsequent to this. So we had an inhibitor that was very potent in an enzyme-based assay. I'm talking about low nanomolar potency. Now, how do we test this further? And I think we had very simple models for testing. We had inhibition of the enzyme, very straightforward. We could do a lot of those. We could measure inhibition of cholesterol synthesis in cells in culture, very straightforward. We could measure inhibition of cholesterol synthesis in the rat. This was just using radio-labeled acetate and following its conversion to cholesterol. Very simple. The compound was very potent in the rat, with ED50s for half maximal inhibition at about 0.2 to 0.3 milligrams per kilogram. However, in the rat, there was no effect on cholesterol levels. Very effective against cholesterol synthesis, did not affect cholesterol levels at all in the rat. This observation was very important to Merck because, well, we had the history at Merck. I want to emphasize the word history of studying this area. We knew compounds like cholestyramine, which worked in people, did not lower cholesterol in rats, but it did have other biological effects. So we knew this. Other companies studying this, and I won't mention names, but 2 companies published that this approach was no good because it did not lower cholesterol in the rat. We had, again, based on our experience with cholestyramine at Merck, and we knew that cholestyramine worked in the dog at lower cholesterol. And it turned out, and I won't show you any data on this today because of time, but that was our key model for studying cholesterol lowering, was the dog. I have to point out, and there's a lot of setting up now concern about what animal models to use. I know compounds don't work in primates and monkeys. It's not, many companies won't take that compound into the clinic. Lovastatin was without any effect in monkeys. It was not absorbed by monkeys, it was absorbed poorly. In mice, it didn't work well because it's very rapidly metabolized. So we were very fortunate. We had models that we knew represented people, at least in terms of cholesterol lowering. And we had an administration, research administration, that was dedicated to the project. So we were able to get into the clinic very fast, and I think this is the history of lovastatin. We discovered this in late '78. By April 1980, we were doing clinical studies in Europe with a natural product isolated from this organism, Aspergillus terreus. I should point out that to this day, lovastatin, Mevacor, is produced by fermentation. It is a very efficient fermentation, and it's where modern molecular biology has played a very big role, because the organism has been modified to a point where all it will make is lovastatin in multi-gram per liter quantities. And it's produced in 50,000 liter fermenters, so you can figure that out. You'll notice that between April 1980, where we first did the clinical studies, you did not have to file an IND in Europe then to do these studies, that we did not submit an IND till March of 1984. Once it was submitted, things moved very rapidly, and there was a reason for this, and I just want to go into it. There was early problems that were, and I say near fatal, because it almost wiped out the program. One was a publication in Japan by Akira Endo describing another compound, monocolin K, which was an inhibitor isolated from a different organism, and it was identical to lovastatin. He filed, we discovered it first, he filed the patent first. So we had in the US, it's the date of discovery that counts. In most of the rest of the world, it's the date patent was filed. So that was one thing that occurred. The second thing was we heard that Sankyo had discontinued their trials on mevacetatin because of rumored tumors in dogs. And then just about the same time that was found in our long-term studies in lovastatin, there were liver changes noted that our toxicologists classified as pre-neoplastic. So we had these issues to deal with. We did not stop the program. We went on with chemical synthesis. In the meantime, there were a few of us that were still dedicated to this molecule and continued our work. Well, I'll just quickly show you how we dealt with the various things. I'll just summarize what happened with the rat liver changes that overcame our concerns. These were mechanism-based changes. They were generic for all HMG-CoA reductase inhibitors. We had a library of them at the time. And most important, these were prevented by the product of the reaction, mevalonate. So mevalonate was fed to the rats. It both prevented and reversed the effect that was seen. And this was a change that was unique to rats. And what it was due to was a big increase in localized increase in the amount of HMG-CoA reductase in the liver. And this was shown by immunochemical techniques, and it was a very clear-cut thing that satisfied everybody and allowed us at least to overcome our fear of the liver tumors in rats. It was very clear-cut studies, and I wish I had the time to go into them, because these were some elegant studies performed by our toxicology group and by our something, by the way, that's not usually done in toxicology groups, that they go into experimental approaches to answer questions. This, at the time, I think, was something quite unique to what Merck was trying to do back in the early 80s. As far as the first item on there, the compound, how to get around the patent problems, we went to chemistry to get around that. And one of the, we tried two approaches. One was total chemical synthesis of novel molecules. We succeeded in that, came up with a compound which seems to be the precursor of we patented it, and it seems to be the precursor for a number of molecules that are now on the market. But we decided that we could move much quicker with chemical modification of lovastatin. We tried the different parts of the molecule. The lactone part up on top was totally unsuccessful modifying that. We could modify the ring system and come up with active compounds, but they were not as effective. The best place turned out to be the side chain. It's a very stable side chain, even though it looks like a simple ester, very stable, doesn't come off readily, but they could be replaced with other groups. A whole series of these molecules were made. I don't know how Joe will appreciate this, but some biologists like to think of medicinal chemists as making compounds. They put methyl groups, ethyl groups, propyl groups, and that one extra methyl group on the side chain. It's an alpha-methyl butyrate. If you take off the CH3, you lose at least 2 logs of activity. Very important methyl group. If you take off the methyl group on the left, on my left-hand side, your left-hand side now, in the ring there, you end up with the compound mevastatin. You lose about 2 to 3-fold activity. We found that the best molecule in our whole series turned out to be the simplest change, and that is simvastatin, shown here. It has 1 extra methyl group in the side chain. Instead of 1 methyl group, you see the downward V, the 2 lines pointing down. There are 2 methyl groups there. That compound against the enzyme, against cells, in the rat, in the dog, and in people is twice as active as lovastatin. Very rare event that you find. You can go from the test tube to people and find the same order of activity. This compound is now the major lipid-lowering agent on the market. It has the biggest market share, and I'll show you why in a minute. And let me just go on. So we had lovastatin, first approved in 1987 in the USA, and simvastatin, first approval in 1998 in Sweden. And both compounds, simvastatin is sold all over the world right now, including the United States. I believe sales of simvastatin run somewhere in the neighborhood of $3 billion a year, and lovastatin about $1 billion a year worldwide. Not bad for a simple natural product and semi-synthetic molecule. Let me just quickly turn to 2 studies. Joe mentioned phase 3, up to phase 3. I'm just going to show you 2, a phase 4 study and a phase 5 study. Phase 4 study was to do the safety and efficacy of lovastatin. If you notice, in 8,245 patients. This started before approval of the drug. It lasted 1 year, and with number of, key thing was the safety, call it tolerability here for the lawyer's sake. Liver function test abnormalities, the concern for what we saw originally, the muscle, which is can be a problem, and lens, because there was some concern initially which turned out not to be a problem. So this is what the Excel study did. It was a very successful study. I'm not going into the details except to say all the criteria were met. And the final study I want to talk about, which was reported in 1994, which is arguably the most important study ever conducted with a lipid-lowering compound. And this was a trial, 4,444 people to look at, the first time, a major study to look for, does lipid lowering really reduce mortality and coronary events? And this took something like, I think, 6 years to perform. Endpoints, mortality. It was a very simple study and design. It was designed to stop when 10% of the patients, 444 patients, died. And that meant they didn't know which group it was from. The study was stopped a little early because in one group there was significantly more patients dying than the other group. Fortunately, it was the placebo group where they were, and this is the results of that study, and it just, what it shows, everything to the left is better, and you can see total mortality, 256 in the placebo, 182 in the simvastatin group. In any event, there was 10,000 to 796, and every parameter looked at, simvastatin was effective. And what it proved is that lowering cholesterol really does decrease the risk of heart disease. The statistics on this were unbelievably good, to the I think 0.0001 level for every event looked at. To end, I want to point out two things. We talk about history. There are people on this list. Jess Huff in the biology and biochemistry, he was the one in my group that did all the animal work, was responsible for the animal work. He was the first to show, 20 years before starting on this, that mevalonate was an intermediate in cholesterol biosynthesis. In the natural product chemistry, Carl Hoffman is on the patent for lovastatin. We share the patent for lovastatin. He's on the patent for discovery of mevalonic acid in the mid-1950s. And we talk about management. The two people that were behind this project all the way were the president of the research lab, Ed Skolnick, who really was at senior management level, made sure that things were happening, NDAs got through, got done on time, etc. And Roy Vagelos, who was the CEO and chairman of the board of the company, who was a prime advocate of this project, and he should have been, since his background was in lipid biochemistry. And with that, I think I better end. Thank you for your attention.
I have a question on when do you file for a patent if it takes 10 to 20 years to develop the molecule as a drug? Is that a tough decision to arrive at?
It is a tough decision. I mean, people play games. We play games. We lost on that one. Now there's patent extension times, and both lovastatin and simvastatin have been given patent extensions based on how long it took to develop. I was involved in one project, which was a failure because of mechanism-based toxicity, but that's beside the point, where we filed a patent early on another natural product inhibitor in the pathway. Turns out Glaxo was working on the same thing. They discovered the compound ahead of us, but they wanted to do all the chemistry around it first. Well, they have the patents on the chemistry. We have the patents on the parent molecule, because they didn't do the work in the States, and we filed first, and we discovered first in the States. So it's a decision. I mean, I don't think anyone could ever think or should think that you're the only person working in this area. We thought we were, We weren't. And people will beat you out on it, and you'll lose the patent. I think, I don't know, maybe Joe wants to.
Just comment on that. You have to realize that the scientists that are working in different companies are highly trained at well-recognized universities. They're reading the same literature, and they're likely to have the same ideas and come up with the same approaches.
Yeah.
So you're in a horse race in almost every area, and you've got to recognize that. So when the discovery is made and verified, we usually file the patent early on, and then the patent clock starts ticking for all the remaining years that we're under development until we launch. So without the patent extension, we probably would have had about 6 years of patent life from the time we launched Felding. We had a 3-year patent extension because of the new laws.
How did you refute the Japanese contention that the same chemical structure caused tumors?
Well, that was a very simple thing. We did the 2-year carcinogenesis studies and did, at humongous doses. And I have a slide showing 180 milligrams per kilogram in dogs, where in people, the top dose that is used is 80 milligrams per person. And we had cholesterol levels that were approaching 0. And there were other problems in these dogs, but there was no tumors noted. I should also point out that what the Japanese did is come back with another compound, which is now marketed. It's known as Pravachol, Pravastatin, marketed by Bristol Myers Squibb. And what that compound is a metabolite of the original compactin, and it was discovered. When we were competing with this competition, and one of my major antagonists is in the place today. We were talking about this earlier. I used to get up and say, well, pravastatin originally isolated from the urine of dogs treated with mevastatin. I mean, that's where it was isolated from. It's not made that way, but that's how it was discovered. So we never found out the true problem there. I've talked to Akira Endo many times. I mean, he's much friendlier to me than anybody at Sankyo, just because by us working in this area, we kept his name up front. And he says they messed up. They didn't know what they were doing. He's not with Sankyo anymore, so he could say it. And he published that. And I have the translations of it. So I don't know what happened there. But they did somehow or other mess up, and we didn't. We capitalized on it because they were ahead of us.
The FDA just didn't. I mean, you didn't.
No, we were concerned at one time because in that time frame when we stopped work on what was then Novinolin, it was resurrected as LOVASTATIN. We didn't change the name, but some organization did. But there were clinical people, lipid specialists out there that knew about our work and knew about the work that we had done in Europe already. And they had patients that they said needed a drug like that. There was nothing around. Then would we give it to them? And Merck's response to that was, well, you're going to have to file your own IND. And I guess a number of us were pretty sure the FDA, who knew about what was going on in Japan, would turn us down, would turn these people down, and they didn't. They allowed everybody that requested the drug to get it. So the FDA seemed to be a lot less concerned about it than we were.
Can we hold questions to the end? Because actually we'll have a brief opportunity to do some group questions, but also I think all the presenters will be staying. So since it is 4:30.